Thank you. Can you guys hear me okay? Okay. I just want to thank the conference organizers for the opportunity to speak, as well as all of the presenters who came before me, because there was a lot of introduction to concepts and foundational aspects of what I'm going to talk about that really set me up well. So hopefully I will also get through this and not keep you from your lunch. I'll also just comment that I can see why people are having problems with this clicker, because it's not straightforward. So Dr. Patel actually just set me up perfectly to start this discussion. He really highlighted how molecules are getting more complex, they're getting more difficult to formulate, they're getting bigger, they're getting less soluble, and they're getting less permeable. At the same time, our clinical trial landscape is accelerating. So for oncology in particular, also for pediatrics and breakthrough therapies, we often see that we're skipping phase 2, going straight to phase 3. And so what that means is that when we start, even at the very beginning before we're in the clinic, it's really important to be thinking about how you're going to take your product all the way through commercialization. Gone are the days when we're just trying to get into the first in human so we can fail quickly. And so now we're really thinking about how can we start with a formulation that is flexible to support clinical development, but also scalable and representative of what people want to see as a commercial product. So we really are looking to get this right from the very start. So clicker first, there we go. So what I wanted to talk about today is how we do this for an enabled drug product, particularly with an amorphous solid dispersion. I put this diagram together to kind of give an overview of the two different parts of drug product development for an enabled product that we might consider. On the left-hand side, we have sort of our enabled intermediate development. And on the right-hand side, we have our drug product. And very often, these two things do not overlap in the middle with a target product profile. They're done completely independently. Someone develops the intermediate, they hand it off to the people who develop the drug product, and you're just stuck with what you have to try to make it work. So what I want to really introduce today is this idea of starting with that target product profile right from the very beginning. So even when you may think that we don't have enough information to define that product profile, I still think it's important to try. And with that in mind, what we can do is develop our intermediates knowing what's coming next so that we're doing a service to the drug product and to the patient to give them something that's acceptable and still has the performance criteria that's needed from the intermediate. So coming back to this idea of a target product profile, I think when we often ask about this in the preclinical space, the answer is, well, we have no idea. We don't know what the dose range is going to be. We don't know what dose strengths we need. We haven't even done a tox study yet. We need to develop an intermediate to get to that point. But it's never too early to start thinking about this. And the part of this is because as we develop our products, they need to be developed with this context in mind. And if we think about the target product profile not as being what we need at the commercial stage, but thinking about it as being something that's adaptable and flexible throughout clinical development, it can be a very valuable tool to frame what we're doing. So some of the things that we hear and some of the things we shouldn't worry about in these early stages are, I don't know what the projected dose range is. Well, that's okay, right? You just projection at the start. If you don't know, you'll probably have some idea of what the range might be. And that's what we should start with at the early stages. And also knowing that that dose range is going to change as you go through your clinical development phases. Same thing with the dose strength. It always changes, and that's okay. But we shouldn't sacrifice the design of our product in the end state for the flexibility that we need early on. There's other ways to go about this where we don't have to make that sacrifice. The drug product form. Oftentimes we start with a solution or a suspension, and then we go to a capsule, and then we go to a tablet. Well, if the tablet is your ultimate preference, why not start with the tablet? Sometimes that's not possible, but in a lot of cases, with the right forethought and the right design, that is an option. And for us, we prefer tablets from the beginning for amorphous dispersions. The size, shape, color, design constraints, if you have these, it's good to know about them from the beginning. We like them to be challenging, and someone yesterday said the circle is king. That may be true for multiparticulates, but we would prefer not to make spherical tablets. But it is important to understand what those constraints are. Those are sort of the program logistics that really can matter. So understanding if you are going to need to coat your product, if it needs to be colored, if it needs to have an embossing or debossing on it. The patient population is important to know from the very beginning as well, right? It may not be relevant to your phase 1 study, but if ultimately this is going to be a pediatric or a geriatric product, if it is going to need to be dosed with the PPIs, we just heard that example, thinking about from the beginning will get you the product that you need at the end without having to do a lot of reformulation along the way. Program logistics. In addition to being more complex and going faster, there's never enough API. So how we strategize the development of a product really needs to take these things into consideration. We can't just assume an ideal situation. So with all of this in mind, in order to actually get a good product early on, right from the start, that can go all the way through sort of your clinical phases of development and your commercial product, as formulators, we need to bring not only that scientific excellence to what we're doing, but also some strategy in order to make this achievable. So going through this, we are working on enabled intermediates, and this is a bioavailability and controlled release conference. I'm focusing on the bioavailability part. We are starting with this API assessment and with technology selection, so I wanted to give a couple examples of this and how we can think about this with respect to the target product profile. So again, someone just went through the BCS classification. I'm sure you're all aware of this and comfortable with these ideas, so I won't spend too much time on it. But what we have on the left is the BCS classification. It's how we think about our APIs and what sort of enabling technologies they might need in order to be bioavailable. On the right-hand side, we've mapped those BCS classifications to a technology selection diagram, which helps us to understand where, if we are in these spaces, where different technologies could be useful for enabling bioavailability. I think what's really important to note in both of these diagrams that sometimes gets lost in the details is that solubility is only valuable as it relates to the dose. So that idea of understanding your projected dose range, even if you don't know exactly what it is, understanding is it low or is it high, is very important even from this early stage to understand whether or not you think you might need solubility enhancement. Because if you have a low dose and you have low solubility, that might be okay. If you have high dose and low solubility, that might be a problem. So in the context of what I'm going to talk about today, which is enabling drug products through the amorphous dispersions, we're really focusing on this low solubility, high permeability class 2 regime. But I think there's a lot of growing interest and need for the high solubility, low solubility, regardless, the low permeability space. And again, we just heard about that from Dr. Patel. One of the things that I'm going to be talking about as we think through how we can actually take an enabled amorphous dispersion as an enabled intermediate and put it into a drug product very effectively and efficiently is really thinking about the design of those two things together. When it comes to adding in permeability enhancement, that concept is exponentially more important, right? We really have to think about when we're formulating for permeation enhancement, we have to think about how the drug product is working in conjunction with the drug product intermediate to enable that permeation enhancement. So it's not the focus of my talk today, but I did want to highlight that it is a growing area, and I think everything I'm talking about today, the concepts behind it are relevant for that space as well. So to give just a little bit of an example on how we might go through using some of these concepts, I have a couple of different molecule examples here. And again, this is probably familiar for a lot of you, but I think that it's a good place to start when considering whether or not you need an enabled intermediate. We are a company that does a lot of spray drying. We really like spray drying. However, if you can avoid it, we would recommend that you do. So if a molecule comes to us as good formulators, it is our goal to understand what is the minimum complexity that you need to get what you're trying to deliver into the clinic. So if you can avoid spray drying by clever formulation techniques, we will help find that because it's a good thing to do. On the other hand, I'll show you why sometimes spray drying makes the life easier of the people downstream from your intermediate formulators. So for molecule 1, you can see the little white spot. I'm going to point to them as I go along. This is a low solubility compound, microgram per mil in both gastric and intestinal media. There's no ionization that's relevant within the physiological space. It's got, moderate permeability. And so it really firmly puts us in that space where we're solubility limited, not permeability limited, and an amorphous form is the obvious choice from a technology selection to get you the boost that you need in order to deliver this molecule. In this case, the only space where their dose would be low enough to not need that would be in the less than a milligram range, and so it's probably not practically relevant. Molecule 2 is an interesting example because this also has very low solubility. It does have some ionization in the relevant range for physiological fluids, so there's a slightly higher solubility at gastric pH than intestinal pH, but it's still very low in both. But what's interesting about this one is that it does have fairly low permeability. And so what that means is that not only do we need to be considering solubility enhancement, but we're kind of right on the edge of needing to consider permeability enhancement. And so what that means is, we're still in that BCS class 2, but you can kind of see where we are in that space. We want to keep an eye on that. If we enhance the solubility, we may no longer be limited there, but we might start seeing limitations in permeability. So it's a good thing to be aware of. The last molecule is a great example of really thinking through the problem statement and understanding the target product profile, the dose range, kind of what's needed for these molecules and who your patient population is. So this is an example where, again, we have moderate permeability, not much of a concern with respect to needing enhancement there, but the solubility is kind of right on that edge. And so we have pretty low solubility at intestinal pH, but we actually have pretty high solubility at gastric pH. And so what that means is that, an amorphous form is most likely going to help here, but you may not need an amorphous form. So something like particle size reduction, maybe a salt form, or just clever formulation of a drug product in which you're leveraging precipitation inhibitors, leveraging acidulants, can really help you kind of boost you into that space where you may not need an amorphous dispersion. And again, this is a great example of this Q value on the lowest row here. That's relating the dose to the solubility. And this is an example of where your Q value kind of crosses over at 8 milligrams or so. So if this is a low dose product, if you're in sort of that 10 to 20 milligram range, you may not need any enablement at all for this molecule. So that's where really understanding what that looks like, what that profile looks like, can be useful. Okay, I'm not going to go through this in great detail, but when you do decide you need an amorphous dispersion, it's also useful to think about how do you select the technology to produce that amorphous dispersion. Spray drying is one option, HME is another, co-precipitation is one that we would all love to do, but it rarely works. Although with some of the more complex molecules that are being produced, this might actually become more of a viable approach in some instances. And so when we think about each of these, thinking about their impact on the drug product design is very important, as well as the performance of the dispersion itself. So we try to consider this in the early stages of screening. Again, we have a whole nother presentation that compares these different technologies, but I did want to highlight that it's important to think about this. So now if we think about kind of this intersection of where the API is being assessed, you've selected a technology, and then you actually have to develop that intermediate. And again, all of you are probably familiar with amorphous dispersions at this point, the ideas that go into actually developing these and screening for the best formulation. And so I'm not going to spend a lot of time on that, and I'm really going to try to frame the time I do spend on that as to how it actually interfaces with the product design for the drug product. But I would be remiss in a bioavailability conference if I didn't talk a little bit about how you develop an amorphous dispersion. So this actually isn't specific to amorphous dispersions, but formulation in general has sort of 3 pillars that we're probably all familiar with: the performance, stability, and processability or manufacturability. I also want to introduce today this 4th pillar, which is design. So when it comes to an amorphous solid dispersion, performance is usually defined by supersaturation and sustainment, maybe the formation of colloidal species, dissolution rate. We produce a variety of different formulations. We do some in vitro testing. What we're really looking for is both seeing if we can achieve and sustain that amorphous solubility enhancement, any apparent solubility that we see above that may be related to colloidal species that we, depending on the molecular properties, can understand if that helps or doesn't help with our exposure. That can translate to in vivo results where we get increased exposure. Stability for amorphous dispersions is always something that needs to be considered, as it does for all products, but there's always a lot of discussion about it for amorphous dispersions. I think at this point, we have a pretty good handle on how to keep them stable and also how to assess stability. This includes both physical and chemical stability. It also includes considerations of both process stability and storage stability. I think we saw some examples yesterday from Bob about how this can go wrong. If you don't consider it early on, you can develop a product that you then later have stability issues for. Processability is as well in the early stages during the formulation screening. We really want to make sure that we have the experience and the knowledge and the awareness to not produce a formulation during screening that isn't amenable to being produced at scale. So that means selection of spray solvents. That means the way in which we're spray drying it. Assuming it's a spray dry dispersion, to make sure that the thermodynamic space is scalable. Again, we had a great example yesterday of a spray solution that wasn't stable. And so, you might be able to get through your early screening with that, but then you have problems when you scale up. So really thinking about these things from the very beginning to make sure that the formulation doesn't just have good performance, but can be a good product. And the design feature is something that's very important to think about as well. And so a lot of times when we're identifying the optimal formulation, as from a formulation screening perspective, what we're looking for is optimal performance that maintains stability that we think we can make. But really, we should also be adding in this factor of that we also think we can put into a product that a patient might want to take and that our program and our clinical colleagues are happy with. And that's the design component. And so the easiest thing to think about when it comes to design is really how much drug can you put in your tablet. And so what's the flexibility in the dose range that you can have that can support your clinical trials, as well as what's the pill burden for the dose that you're trying to target. And one of the things that's important when we think about selecting our optimal formulation in an ASD screen, it's not just the amount of drug that's in the SDD. It's also what SDD formulation you're selecting, what polymer you're using. And part of this is because when it comes to a tablet or a capsule formulation, enteric polymers have less of a tendency to gel, which means it is easier to put higher amounts of SDD in the tablet. You're more limited with neutral polymers. We do have some clever techniques that allow us to get to 30-40 weight% of an SDD for a neutral polymer, but it can be very challenging. And so all things being equal, if there's only a small difference in performance between, say, your HPMC formulation and your HPMCAS formulation, even though the HPMC maybe gives you 5% better bioavailability, you might be able to only dose half the dose strength in your tablet. So you might want to consider the HPMCAS being the one that you put forward if you're thinking about design comprehensively. So we'll talk a little bit more about this as we go into some of the next slides. So once we've selected that formulation, hopefully we've thought about design as part of it, then we're thinking about the product design and the formulation screening for the drug product. And I've been trying to use the word drug product and not tablet because it doesn't have to be a tablet, but most often people want tablets, and we actually, for a variety of reasons, prefer tablets for our amorphous dispersions. One reason is because we have more control over the disintegration mechanisms. Actually, we heard a great talk by Prajwal yesterday, which will come in again and again as I kind of go through this. And because of the nature of gelation of those polymers, having more control over the disintegration where we don't have to wait for that capsule to disintegrate first helps us to be able to produce a better drug product. The loading is another thing, right? Tablets have less of a pill burden than capsules typically, so we prefer them. We also prefer them because we kind of grew our systems during COVID when we couldn't get capsules. And so now we're just as fast and just as efficient with just as little material to develop tablets because we had to be. So what I really wanted to introduce today is this idea of using a tablet platform for our amorphous dispersions. And so like I said before, most people want to avoid a spray dry dispersion or an amorphous dispersion if at all possible. It adds more complexity, takes more time, it takes more material, it costs more. And all of those things may be true. But the people who really prefer to use an amorphous dispersion are the tablet formulators or the drug product development team. Because it doesn't matter what API goes in there. The spray dry dispersion takes on the properties of the polymer and the particles. And we can control the particles through the spray drying process. And there's only a handful of polymers that are typically used in these formulations. And so we know what those properties are. What that means is that it's actually very simple, very fast, very robust to develop tablets with these formulations. And so you can save a lot of the time, a lot of the complexity, and a lot of the risk by actually selecting an ASD formulation and making it into a tablet. I think, interestingly enough, when we think about these platform formulations, we often talk about crystalline formulations that don't use ASDs as conventional. I really hate that term, conventional, because it suggests it should be easy. I think the most difficult tablet formulations that we have to develop tend to be crystalline products because you still have the same criteria. You're moving really fast, you have little material, and then all of a sudden you have an API that changes with every lot. And so it's very hard to develop a robust process that actually could be scalable through the entire phases of clinical development. Not that it can't be done, but it's much harder. So I wanted to introduce this concept of a tablet platform and kind of give you a sense of what that looks like and how it performs. So there's three parts to the platform concept. There's the process train. So we have several different unit operations, and I'll get a little bit more into kind of where they fit in. But they're all based on using roller compaction as a dry granulation process. We always have an extra granular addition. And then we have compression using a rotary press typically, and the film coating is optional. Depending on, again, your target product profile, your need, where you are in the clinical phases of development, you can either choose to coat these or not coat these. The tablet composition is also a key part of this platform. Now, we have several components that we always have in these formulations, and I'm not going to get into the exact details because we can't give away all of our secrets, but we do have preferred components for each of these that actually change based on which SDD polymer you're selecting. So we have preferred ductile and brittle fillers. I will say that our preferred filler, one of our preferred fillers is the Avicel, for all the reasons that we saw yesterday as far as how it supports disintegration and water ingress. We have preferred disintegrant. The preferred disintegrant does pair up with the different SDD polymers, so they don't all work equally well together. So knowing which ones match well is important. And then, of course, flow aids and lubricants are also part of this. And the last part that really makes this enabled as a platform is the tablet architecture. And so I think Prajul yesterday gave a great foundation to understanding why tablet architecture can matter. And it has to do with, at the micro and nano scale of the materials we select and the way that they fit together, how water ingress and disintegration act in conjunction to be able to break apart that tablet, get it back down to the granules, then from the granules, get it back down to the primary particles so that your SDD can be released for performance, for adequate performance the way it would be without being impeded by the tablet at all. And the last part is really thinking about how we can take this platform and support that idea of dose flexibility. And so again, there's this perception that in order to get dose flexibility early in your clinical trials, you need to have powder and capsule or you need to have a powder and bottle. And that certainly gives you dose flexibility, and sometimes that is important, right? Sometimes you need the dose flexibility at the bedside. But more often than not, you just need dose flexibility up until the last minute before you have to make materials that then have to go into the study, but you don't need sort of infinite flexibility. So what that means is that we need to think about this in the development process to be able to support a very wide range of doses that can be selected for use not just in your first clinical trial, but as you progress as well, and ultimately can be refined for your commercial dose strengths. And so this, again, it depends on the polymer you're selecting in your SDD, and it also depends on the drug loading. So I just gave a couple examples here. But we can, with 2 common granulations, we can really cover a very broad range of doses. And if you wanted to go beyond that, even more common granulations could be considered as well. But 2 usually covers sort of the broadest range that you're going to get. And so for an example, with an enteric polymer where you have 25% of your API is in your SDD, you can get up to 50 or 60% of that SDD in a tablet pretty comfortably in our platform formulation. We feel confident telling you you could do that. I gave 10% as the example here on the lower end, but honestly, you could go down to 3, 4, 5%, and you'd still be in a pretty comfortable space where we wouldn't feel like we needed to do a lot of development work to demonstrate that that's feasible. That can get you, between 2.5 to 150 milligram dose strength quite easily. And then all you're doing in between there is changing the size of the tablet. The neutral polymers, like I said, they're a little bit more limited. We can't get quite as much SDD into the tablet. And so again, when you're thinking about selecting the drug loading and the polymer for the SDD, that needs to be taken into consideration. But we can get to 30%, maybe 40%, depending on the properties of the polymer or the properties of the API. But that's one that we need to test a little bit more. So our platform formulation, again, it's a maybe more considered a platform approach, consists of these key components. We know what works, we know what doesn't work, and I want to try to convince you of how we're also comfortable with respect to not just the performance of the tablets, but also the manufacturing and the scalability and processability of these platform formulations. So again, someone very nicely introduced both the concept of disintegration being a very key factor for tablets, particularly for an immediate release ASD tablet. The goal is to deliver that ASD, the primary particles, to the GI tract without having introduced any harm to them from the tableting process or from the tablet disintegration process. So we want rapid disintegration that releases those particles. I think yesterday we heard that a lot about how friability, you don't have a product unless you have adequate friability, right? It doesn't matter what kind of equipment you have. If your tablet falls apart in it, you can't overcome that with equipment modification. Same thing with your bottles. If your tablet's too soft or too friable, it will be destroyed in your packaging or during packaging. So these are the 2, they're not the only, but they're 2 key product attributes that are critical to tablet performance. So when we think about how we actually control these, it's usually through tensile strength or hardness. We prefer tensile strength, and you'll see for the purposes of this why we use that as a scalable factor, because tensile strength is basically the hardness measurement that's been normalized for the tablet geometry. So what that allows us to do, and you can see it in this example here, is to normalize across different strengths. So this is the same formulation, same product, but it's just 2 different size tablets. And when we normalize for tensile strength rather than hardness, we can see that these kind of collapse onto a single trend. So what we can see here, and this is not unexpected, is that friability, as you increase tensile strength, your friability goes down. And lower friability is better. At the same time, when we increase our tensile strength, disintegration goes up. And so, for an immediate release product, slower or faster disintegration, so lower disintegration times is preferred. And so the key is in a platform or in any given formulation, can you find a space where these 2 things overlap, that you can both produce a product that has acceptable friability and acceptable disintegration that you can manufacture robustly? So when we look at this across a wide variety of different products, different strengths, different SDDs, all within our sort of platform for this HPMCAS SDD tablets, you can see that the first thing to point out is that we see very similar trends in disintegration and friability with respect to all these different products as they relate to tensile strength, regardless of the amount of SDD that's in the tablet or the amount of drug that's in the SDD. And we see that there's a pretty comfortable space where we are getting both our friability below our sort of upper limit for what we target. And we typically target a 0.3%, which is kind of well below the USP limit, but we want to be conservative here. And for disintegration, I don't think there's a hard cutoff, but for an immediate release tablet, again, to be in a robust space, we're putting a target at probably 5 minutes or less. You'll see there's a few points that fall above that. There's actually just one product in here that was a kind of a bad actor for disintegration, but it's still kind of within an acceptable range for an immediate release tablet. But the majority of these fall into a nice, comfortable space where if we're somewhere between 1.5 and 2.5 millipascals for our tensile strength, we can be confident with this formulation platform that we're going to get good friability and we're going to get good disintegration. So that gives us confidence that we don't have to do a lot of screening when it comes to the tablet formulation to be able to achieve these metrics, which means we still always want to confirm it, right? I'm not suggesting that we wouldn't do any development work, but we can kind of plug it into these systems. We can design the tablet based on the dose strengths, the amount of SDD that goes into the granulation. Again, that's based on the dose strengths. So not only do we need to be able to make tablets that perform well, but we also need to be able to make them in processes that are scalable. Because again, the idea here is that you go into phase 1, you have this flexible platform formulation that you can get a bunch of different strengths. If it looks good and you're fast-tracked, you might be making phase 3 batches in a year. And so there's not a lot of time to reformulate. If you don't want to have to reformulate, whether it's for performance reasons or for processing reasons, in order to get a scalable and robust and stable formulation. And so I can't go through all of the details of scale-up, but I did want to give a couple examples of how we think about these concepts, and then also how it translates to this platform that we're talking about. So again, we talked about our product attributes with friability and disintegration being very important for our tablets. We then talked about how tensile strength is sort of that scalable property that we're going to go after, regardless of whether we're making these on the bench scale or we're making them on a rotary press or we're making them on a 8-station versus 32-station rotary press. We're dialing in the tensile strength or the hardness for any given product in order to target that desired range for friability and disintegration. And so when we think about the process parameter that we're using in real time on the floor in order to do that, it's compression stress. So if we look at that, we typically, when we're assessing these formulations early on, are using these CTC profiles, which is compactability, tabletability, and compressibility, and I'm going to do them out of order. But in order to understand with this specific product or the specific platform, where are we in that manufacturing space based on these material properties, and do we have enough sensitivity and robustness in order to have a scalable manufacturing process. So the first one is compressibility. And so again, here, what we're looking at is actually solid fraction, which was introduced by Prajul yesterday. It's the inverse of density. So solid fraction of the tablets versus the compression stress. So this is really easy to visualize. As you compress your tablets, you're removing air. You're removing the void space. And so your solid fraction is going up. And we saw that with the same formulation, in Prajul's talk yesterday, that with the same formulation, as you increase that solid fraction and you remove that void space, the percolation goes down. And so therefore your disintegration goes up. And so what we can see here is this similar trend, right? Solid fraction, as you increase compression stress, your solid fraction will go up, but with sort of decreasing return on investment, right? It's not a linear function, and we wouldn't expect it to be. And ultimately, you're never going to get to that solid fraction of 1 in any sort of reasonable stress for a tablet press that we might have. And again, that was really well illustrated yesterday because some of that void fraction is on the nanoscale. You'd have to completely compress your materials and break all of the architecture of not just your tablet architecture, but your excipient architecture in order to completely remove that space. So that's not the goal. Our goal is not to be in that plateau region, right? Our goal is to be in a region where we can comfortably manufacture and hit a solid fraction that gives us our product attributes. So in order to think about that, we then need to translate tensile strength to solid fraction. And if you look at these plots, it looks like, okay, everything falls on a similar trend. But again, that's because this is a platform formulation. So it's not guaranteed that these relationships always look like this. But in this case, we can see that if we're trying to hit our tensile strength between 1.5 and 2.5, which is what we just talked about in the previous slide, we're looking at a solid fraction that's somewhere around 0.8. And so if we combine these two things together, we can get our tabletability profile, which tells us in order to get the tensile strength sort of in the range that we're looking at or we're looking for, what are the compression stresses that we need to be able to do that, and are they in a range that is amenable to our manufacturing process. And so you can see with this platform that we are, we don't typically want to have to be above 200 millipascals in compression stress. That's a good upper range for our rotary presses. So we can see that we can comfortably get to the tensile strengths that we need with well below that 200 millipascal range, but we also can see that this relationship isn't too steep. So if this relationship, if our tabletability was very, very sensitive and the slope was very, very steep, it would actually be very difficult to get your target tensile strength without a huge amount of variability, right? Because if you have such sensitivity to that compression stress, then just normal variation in the process might result in high variability in your product. And so looking at this, it doesn't mean that you don't have to identify narrower windows as far as the targets that you're going after for the manufacturer, but it gives us confidence that this platform formulation is going to work. You're going to be able to dial in those parameters. These features of the material properties, they depend on a lot of different things. They depend on the formulation, they depend on the ingoing material, they depend on the particle size or the granule size. All of these things have normal variations from batch to batch and lot to lot. So we want to make sure that we're in a range where we can actually dial in that compression stress for any given batch or any given formulation or any given lot so that we can hit the target tensile strength, which is kind of our key parameter here. So hopefully that gives you an idea of how we think about product design right from the very beginning of our ASD formulation screening and thinking about that target product profile, which really brings these two aspects of development together and makes it really a patient-centric approach to delivering something that works for the program strategy as well as the clinical strategy, and ultimately gives us a way to go very quickly from first in human through commercialization without having to do a lot of changes with respect to formulation or process. And so I just want to spend a couple minutes on the concept of process scale-up, because a lot of what I just showed is actually done or can be done with bench scale techniques. But there's still a need to scale up. And actually with our platform formulation, because we've done this so many times, and we just saw the CTC profiles on the previous slide, which give us that confidence as far as the design space for manufacturing, being robust and having enough flexibility, oftentimes the scale up can be pretty minimal. And it's actually usually driven by the need to produce materials for stability studies over actually needing to demonstrate or develop the process, because again, we have that's sort of tuned into how we think about scaling. So this is an example of the process train that we might do at bench scale. And so we might blend by hand or with a turbula. We use a compaction simulator and a Gerteis hand mill in order to do our dry granulation or simulate our dry granulation at the bench scale. And then we again use the compaction simulator in order to produce the types of CTC profiles that we saw on the previous slide. This again allows us to do all the development with grams of material rather than hundreds of grams of material. I will say that one key aspect of this, again, I mentioned on the previous slide, but particle size, both the STD particle size, the excipient particle size, the granulation particle size, those things are critical for understanding those CTC profiles. So if those things change, that CTC profile will change. And so what that means is that in order to do this bench scale work, you need STD that's representative of a large scale STD process. And we are fortunate that we have those systems at the small scale so we can match them together and produce STD that has the particle size you would see on a large scale using sort of milligram scale quantities if we need to. But it is a key part of this. The milling is another great example of this. Gerteis actually, we prefer the Gerteis systems. They sell a hand mill that allows you to actually mill ribbons or mill slugs that using a mill that is representative of what they have on their actual at-scale systems. And so we end up with particle size distributions and granulation properties that are representative of the process scale. So with all of that in mind, we can then, because of this material that we've used at this bench scale and produced those CTC profiles and identified what our targets are, we can use that information to sort of rapidly scale based on those material properties. And so I talked mostly about this compression step, right, and how we scale that based on tensile strength and how we use those CTC profiles to be confident that we're not on the edge of failure. I think Feroz yesterday also highlighted that if you're on, it doesn't matter how good you are at scale up, if you start on the edge of failure, you're gonna have problems. And so our whole goal in this is from the very, very beginning, making sure we're not on an edge of failure. And using our platform formulations, we can confidently make sure we're not on that edge of failure and go very quickly without actually having to do a lot of process development work. So the same concepts apply for the other unit operations. I didn't talk at all about granulation. In granulation, again, similar concepts, but we're using the solid fraction of the ribbon rather than tensile strength. In part, you already saw the relationship between solid fraction and tensile strength. For ribbons and granulation process, it's very difficult to measure tensile strength. That's why we use the ribbon solid fraction as a scaling parameter instead. And of course, for milling, we can't forget that that's actually not a simple thing to scale up, and so we use Freud numbers for that as well. So in conclusion, I think there is an increasing need for more effective and efficient development of scalable formulations that can start with phase 1 and really that you can envision going all the way through your commercial phase very quickly, right? I think that there's a lot of examples of sort of the accelerated pace of clinical development that we need to support. The ASD technology selection and formulation development, again, I think that's actually been pretty well sorted out in the industry, right? There's a lot of examples. These conferences used to be filled with how do you develop an SDD? How do you decide what the right formulation is? And we've kind of moved past that in that there's a pretty standard approach to that. Doesn't mean it's easy, but there's a standard approach. But really what we need to now think about is how do you wed that intermediate development with the final product development so that you can get through the whole process very efficiently and have the right formulation right from the very start. And what we found is that platform formulations for these amorphous dispersions really enable rapid material sparing and scalable product development. Not every formulation works, and I think sometimes it's very challenging to develop STD tablets, but what we've seen is that it actually, over the years and with a lot of experience, we almost always select the same formulation to the point where we can call it a platform. It enables a wide range of loading. The properties that we care about when it comes to these drug products, they don't depend on the amount of STD that's in the tablet, they don't depend on the amount of drug that's in the STD, and they don't depend on the properties of the molecule that's in the STD. They just depend on the nature of the particles that we're producing, which we, again, engineer specifically for this purpose. These CTC profiles, having suitable CTC profiles ensures robust operating space, gives us that confidence that we'll be able to easily scale this. We continue to use those profiles throughout the scaling process as well to understand how different features of the ingoing materials or how things like dwell time or different features of different rotary presses might impact those relationships. And those scalable parameters really are what enable sort of that rapid and easy scale up. So with that, I hopefully am on time and not holding everyone up from lunch, but I'll take any questions, or I can take questions outside of the room as well.
Right. First of all, let's give Erica a round of applause. That was a wonderful talk. So thank you, obviously, to Erica, and alongside Erica, we've got Rod, Gerard, and Lauren that are in attendance as well, if you have any questions specifically for the Seran folks. But anyone specific for Erica right now, just based off of her talk before we break for lunch? So, let's see. We've got maybe more. Yes.
Excellent talk. I think I really like the idea of developing a tablet ASD right from the beginning, right? And it can support all the way beyond the phase 1. One question is that tablet is usually preferred, and we like tablet. But under which circumstances would you have to consider capsule formulation? Do you encounter any kind of cases like that?
Yeah, it's a good question. I think my first answer is probably going to be a little bit of a surprise, but the most common conversations we have about capsules tend to be just based on people's assumptions and perceptions that capsules are going to be faster, cheaper, and easier. So usually that's where the conversation starts. I want capsules because it's easier. And we're like, well, it's actually not easier in this case. It's actually more difficult. So we can develop capsule formulations using the same principles we use for tablets, but there's more chance it won't work than the tablet. That's for the SDD formulations, though. I will say that when it comes to conventional products, capsules oftentimes are faster and simpler than the tablets because you don't have to worry about sticking of the material, and you don't always have to worry about flow properties as much. So that's where the API properties for crystalline products can really get you. So the idea of tablets being preferred and kind of being the go-to and being even simpler and easier than capsules, that's really for this SDD platform technology.
So when it comes to clinical trial, people go active in capsule, yes? That's the first thing that you do. You just immediately put the active in the capsule, go to a phase 1.
If we could only all be so lucky.
Yeah, so, but I mean, ASD is similar, I had seen similar concept, which was SDD in the capsule directly, going into clinic without developing anything. So you want to skip that and, just go all the way to tablet then.
Yeah.
That's...
And I'm not saying that you can't do that, right? Like sometimes people are still doing that, but there's a couple of reasons to consider not doing that. One is that actually putting SDD in a capsule is very challenging. If you're doing it, especially at the pharmacy, it's exceptionally challenging. If you're doing it with processing equipment in the suites, I'd argue why not have a tablet, right? You're already doing a GMP manufacture. But the other aspect is that SDDs in capsules often don't work very well. They don't disintegrate very well. And so if you have a very low dose and a very large capsule, sometimes it can work. But a lot of times if you're trying to pack SDD into the capsule, you end up with sort of gelation during that disintegration process that can inhibit the performance. And that doesn't mean for every molecule, right? But for a lot of them, it can be a challenge. And you can't scale quickly, right? You then have to do a bridging study to get to a product that can actually get you into sort of that phase 2, phase 3 space.
Hi, Erica. One question, since you spoke about a little bit on permeability. Firstly, great talk. I love the idea of connecting ASD to have it into a product. So what's your experience using permeation enhancers in spray drying? And would you prefer in ASD or would you prefer as in the tablet, in the product?
Yeah, that's a really good question, and it's something that we are actively looking at and interested in. And I think that there's not a straightforward answer as to where is it better placed. And part of it is going to be dependent on the permeation enhancer and the molecule. And so, one consideration is this goes back to, I think, the industry as a whole also doesn't always understand how these two components act together to enhance permeability. So if the goal is to have intimate mixing of the molecule and the permeation enhancer so that they can form a complex, and that's the mechanism by which you're kind of getting to the epithelium at the same time and enabling permeation enhancement, then a molecular dispersion of those two things together could be very enabling. Right? More so than just mixing them together as a physical mixture in the tablets. But if your goal is not to get them intimately mixed in order to form a complex, and you're more focused on just delivering them to the site, to the epithelium at the same location at the same time, then it may not matter if they're in the SDD or they're in the tablet, but the design of the tablet and the intermediate really matters, right? You need them to dissolve at the same time. Ideally, they're dissolving close to the site of action. So if you think about the tablet architecture and the solubility and the dissolution rates of the permeation enhancer relative to the API, it becomes very complex very quickly. So again, I don't think we know, but then the third aspect to consider is, if you look at the success of permeation enhancers, particularly in the peptide space, it's huge amounts of permeation enhancer for small amounts of drug. And so depending on, again, the properties of that permeation enhancer, you may be limited to how much you can get into the STD or how much you can get into the tablet as a physical mixture, because they're not the easiest materials to work with. So those are the types of things that I think we as a company, but we also as an industry, are still figuring out.
Yeah, we had a poster at Evonik that I worked on years back, and we made Calitra without permeability enhancer, and human data was showing absolutely zero bioavailability for the product that is in the market. And then we made with the O-Irrigate E without that, and it worked well. So there's a poster that we have on that front, but you are right that depending upon what you are looking for, you have to decide where you want to put it, inside the ASD or outside.
Yeah. And one more comment on that. I think we started this conference by saying, for modified release, right? So not permeate, but the same holds for permeation enhancement for modified release. You're looking at those, BCI, the high solubility compounds, and you're saying these are the ones that you can formulate well, whether it's like high solubility, high permeability for controlled release, or it's high solubility, low permeability for using permeation enhancers. Like those are the ones that fit into those concepts. We actually are interested also in, that's great. I mean, we would all love to have high solubility compounds, but sometimes that's not what we get. So how do we actually think about supporting controlled release and modified release or permeation enhancement when you also have to enable the API for solubility enhancement? And so that adds one more layer of complexity, which again, you just can't ignore the drug product part of that, because the goal is not just to deliver the intermediate. It has to actually act in conjunction with the excipients in the drug product. So.
Perfect.
Okay, let's give Erica a round of applause. Thank you.